So welcome everyone to the panel discussion this afternoon. I know it's late in the day and especially for you guys on the East Coast, so appreciate you hanging around. It's going to be an incredible panel of accomplished scientists. They're not only accomplished scientists, but they're very enthusiastic about their work. We've had some great discussions before today, and I know that everyone will enjoy it. We're going to get a great perspective from the panelists since we have representation from academia, a charitable foundation, small biotech and small pharma, a software company, other service providers, as well as big pharma. So I'm Tina Rogers. I'm a preclinical toxicologist. I've spent my career in preclinical CROs. I'm currently with Sinclair Research Center as a senior scientific advisor. Sinclair is a full-service preclinical CRO located in Missouri. So what I want to do is let's start with some introductions by the panelists. And so please mention your role and your company and how your role relates to drug discovery. So let's start with Jackie.
So I'm Jackie Salm. I'm the Chief Scientific Officer and co-founder at Celera Bioscience. We are a small startup biotech in the psychedelic space, but most of my background was in natural products and small molecule drug discovery.
Great. Yeah, we heard a great talk today from your CEO.
Yeah, Chris gave a great talk.
Okay, Eric?
Hi, I'm Eric Martin, and I work for Novartis, and I'm a computational chemist doing early-stage drug discovery.
Zhen?
Hi, I'm Sheng Yi. I'm a manager in the discovery research team at the Genevant, which is a small biotech company located in Vancouver, Canada. And our company focuses on RNA delivery, including like lipid nanoparticles and the bioconjugation. Actually, our RMP is involved in the first approved siRNA drug on Patro in the history, and also it is involved in the current battling effort against COVID-19. And also, our siRNA conjugate platform has demonstrated clinical safety and efficacy. And I was trained as a molecular and cellular biologist and have accumulated some experience in biology, liver disease, and RNA therapeutics. And I'd like to thank the organizer and for this meeting to have a discussion today with a great group of scientists. Thanks.
Great. Woody?
Hey, Woody Sherman, Chief Scientific Officer at Silicon Therapeutics, and we're now part of Roivant. I'm also the Chief Computational Scientist at Roivant, and focuses primarily on building computational tools, primarily physics-based tools, but also AI, machine learning, data science tools to overcome critical challenges in drug discovery.
Great. Rami?
Hi, everyone. I'm Rami. I'm the in vivo scientist at Juvenile Therapeutics. We are developing protein-based therapeutics to treat age-related disease. And my work focuses basically on developing in vivo models to support the efficacy of our lead therapeutics.
Okay, Whitney?
Hi, everyone. I think based on the screen display, I'm part of the Brady Bunch, the way it looks here. Otherwise, my name is Whitney Smith. I am a business development manager here at Collaborative Drug Discovery, which means I'm part of the sales team, really. And so, my background is as a medicinal chemist, kind of moved into cheminformatics and then ended up working with you fine folks to try to solve the hard problems around data management. Love it.
Great. Jeff?
Hi, I'm Jeff Dietz. I'm an associate professor at UCSF in the pharmaceutical chemistry department. I spent the first 10 years of my career in biotech, working at Alon Pharmaceuticals in the theater neurosciences, and have been at UCSF now for 10 years. And I also co-direct the Small Molecule Discovery Center, which is dedicated towards discovery of new therapeutics.
Great. Manish?
Hi, everyone. So I'm Manish Pingle. I head the business development team for discovery services at Sci Life Sciences. Sci is a full-service CRO that does discovery, development, and manufacturing. We work with companies that are represented here as well in trying to help them get their science forward. Trained as a medicinal chemist, spent some time in academia, spun a couple of companies out, worked with them and then I moved to the business development side for CRO. So it's good to be here.
Okay, great. So I think we're missing Letitia, unless she's somewhere I can't see her.
Yeah, Letitia has an internet problem, I believe, and also a power problem. She sent an email to Sherry saying that her neighbor has issues. Yeah.
Okay. All right. Well, thanks very much. So I'm going to attempt to share my screen with a slide. Just to help frame our discussion today. Can you guys see that?
Yep.
Yes.
Okay, great. So actually this diagram is adapted from a similar one on the Tau Consortium website. So it was courtesy of Letitia. So I'm sorry that she can't be here to see this. So this basically is what we're talking about today. We're talking about emerging strategies to ensure the success from drug discovery to drug development. And we'll be talking about different aspects of this graphic here. So I pretty much live down here on the right-hand side, pretty much exclusively, and we have representation from everyone who hits at some point on this graph. So I'm going to stop sharing now and get back to the discussion. Okay, so we had a couple of meetings before the panel just to try to come up with some themes that we wanted to discuss today. And so some of those were, included the fact that we need interdisciplinary approaches to help with this drug development process. We need to leverage AI. We need to maximally utilize our data. We need to have a deeper understanding of our disease models. And we need systematic processes to help us take IP, for example, from academia to the clinic. And also that we need customized approaches because every drug is different. And even getting into marketing and competitors and patient populations and those kind of things are critical considerations, which is we don't really consider part of the process of drug discovery. But why don't we start with the interdisciplinary approaches that are needed? Eric, I know that you made some comments about the changes in the role of a medicinal chemist, even 10 years ago, and how different that is now.
I think even 10 years ago, it was just starting that the most important thing for a medicinal chemist was to be an excellent at synthesis. That's still obviously extremely important, but increasingly medicinal chemists are having to understand PK, having to understand tox, having to understand computational chemistry and molecular modeling tools. And molecular modeling tools are also coming to meet them halfway, and tools that used to be very complicated and detailed, giving the user as much control as possible, have now been turned into workflows that can do many of the things that had been done by hand automatically. And I think that, and I think that's true also of all of the other specialties. Drug discovery has always been extremely interdisciplinary, but I think increasingly, instead of a bunch of experts in their individual disciplines getting together to solve problems, that increasingly it's been important for people to learn ever more of the science and the vocabulary of each other's disciplines, and to have tools available to allow people to work outside of their main expertise. For example,
Jeff, I'm sorry. Go ahead.
For example, I'm a computational chemist working with synthesis chemists. I used to really rely on them for synthesis, and at the present I still do, but there are increasingly sophisticated retrosynthetic tools that would be making it so that I could meet them halfway in terms of more plausible proposed compounds.
Great. And Jeff, do you have some comments about this? About interdisciplinary
I do. I come at it
approaches?
from a very similar point of view where, Eric talks about these kind of extensions of knowledge. And I think of it kind of like in the past, we would fit together like jigsaw pieces with very sharp edges. In chemistry and biology, we knew each other, we interacted with each other, but there were boundaries. And I think over the last 10 years, it's gotten way more frayed. So the edges really intertwine more intimately, and more is expected of each other. I expect biologists to know a bit of chemistry and know what an amide bond is. And they expect me to know what a protease is and know the difference between an aspartoprotease and a threonine protease. And that's a good thing. I think the more common language we have to work together, the better shot we have to do something really special. And that's kind of been the name of the game. The easy targets are gone. The hard targets are left. And it takes people who really know what they're doing and know how to work together to ferret these things out. As a synthetic chemist, as a trained chemist, I need to know biophysics. There's no ifs, ands, or buts. You just got to know it. And it's the reality. I think it's a good thing. It's a very good thing.
Anyone else have a comment about this? About interdisciplinary approaches?
Yeah, I just have
it's essential.
Yeah, I just have a quick comment regarding to previously, our department was called biology department, and our new CSO come in. It's like, why do you call yourself biologist? You are not biologist. All you do is drug discovery, and drug discovery takes all the different expertise you need: chemistry, toxicology, PKBD, biology, you name it. So you should not call yourself a biologist. Just call yourself discovery research. So we changed our department name to discovery research and been using. And then I think this really relate to the theme today. Is it's gonna be interdisciplinary from day one, and also not just chemistry and biology, but also business, economics, right, and IP, all laws. And also logistics, you name it. You just need to understand the ins and outs of everything. You may not be the expert of everything, but as a manager of a project, you need to have dive into all the different disciplines and know how to gather the right people together and form a great team to make things happen. So in that way, you definitely need a lot of knowledge on different area for sure. So.
Right. And, I remember Jeff made the comment in our call about that drug discovery doesn't exist really as a discipline or as a curriculum in PhD programs. And, at least back when I was in school, your success was all about you, and nobody taught you how to work with other people and work with teams. And that just becomes so clear when you start working in drug discovery. So I'm going to stop there.
That's changed a little bit, I think, over the years, too, because it was true when I was getting my PhD, and we're going back 20 years now, that there were some courses you could take. There was a medicinal chemist or two, and I was at University of Michigan, where John Topless had recently retired. And he was kind of known for this Topless tree and a way of systematically doing these types of molecular optimizations. And there was that kernel of a beginning of that. And in that intervening time, now at UCSF, we have several classes that are not only on optimizing stuff like John used to teach, but also how to do drug discovery, how to do target validation. I teach a seminar every spring about this, and I so wish I had this 20 years ago, because it really gives you a glimpse about what you're getting into when you start thinking about a career in drug discovery, to know these hundreds of people that are going to work on this team in the hopes of getting something into a person at the end of this long journey. So I think it is changing. I think there's the beginning of that being taught in some schools. And of course, UCSF is very kind of biomed forward, right? The chemistry side, I think, is kind of a tertiary element in elevating target biology and kind of the screening side of things.
Okay, I'm going to stop there because Letitia has actually joined us. And so we're going to give her a chance to introduce herself and just talk about your organization and your role in drug discovery, Letitia.
Great. And apologies, I had a transformer blow up. It was a really scary thing just outside my house. So no Wi-Fi. So I'm Leticia Sherman. I work at Charitable Foundation that funds the program Tau Consortium. It's a consortium of mostly academic investigators that are trying to research tauopathies. These are about Alzheimer's-related dementias. And my role at the foundation is to take drug discovery programs from academic setting all through the valley to where they have strong proof of concept, solid drug discovery along the way, so that they can be partnered off through partnerships with pharma or biotech, or be licensed to pharma, or to actually get VC funding. So that's kind of my role, and it's in many ways it's a didactic process of going through the process of making sure that every step of the way the data is robust, that we have strong foundational way, that all the data that represents these packages are really up to standard to be to go through scrutiny through to a funder.
Okay. So we were just talking about the interdisciplinary approach as one of our themes. So anybody have any other comments about that and how important interdisciplinary approaches are, or a different take on that, or how your organization handles it? Jin told us that his department got a new name, but anyway, beyond that.
I have a thought, which is something that we face often, as a software provider for chemistry and biology and all the various subdisciplines, is that, different disciplines look at data differently. An extreme example is people will talk about a chemistry ELN and a biology ELN, and they tend to have even oftentimes when they're on the same platform, they tend to have different functionality, different buttons necessary for the different operations that each of those disciplines perform. Sometimes you can have all that in one application, and sometimes you can't. It really depends upon the environment, depends upon the type of work folks are doing, depends upon how they want to see their data and what they want to integrate it with. So, as we are joining, because I agree with everything that everybody's been saying here, which is, we've gone from these very sort of separate things to a much more interconnected drug discovery effort. And one of the real challenges, something I brought up during my talk, is that the industry does have to come to terms with common frameworks, common languages, common standards, so that we can provide tools that actually talk to each other. So, you don't end up with a bunch of different disconnected systems and silos of data that end up in dead ends. And it can be very difficult, and it can be very challenging, but at the end of the day, it's really rewarding when you see a company execute that really correctly, and you can see a real benefit.
Yeah.
In discovery work.
Yeah. We just went through an exercise evaluating ELNs for molecular biology workflow, chemistry workflow, and kind of protein production workflow. And you're so right. It's really, really, really hard to find one that does everything well. And it's not even the data you collect so much as the workflow that you do, where you might be a month-long optimizing protein production, whereas a chemistry experiment is probably a day and a half most, in terms of running a reaction, do the NMR, and move on to the next step. And it's just finding that common framework for us all to work together was a major goal of that project. And I did evaluate CDD's notebook for that, and it performed very well.
I was going to say Whitney will sell you one.
Oh, he did. I'm a huge CDD fan. I'll put that disclaimer out there.
Yeah.
Jeff and I have a coffee shop we go to there in that little breezeway.
Yeah.
I have an empty sandbox somewhere in CDD as well.
So I've come into what I think is kind of an example of that, is where, as I mentioned in my talk, I'm currently doing massively multitask modeling, where for each compound I make predictions on 8,000 different assays. And so now comes the time when I want to be able to say, okay, given these assays, which ones have similar SAR? Can you make SAR omics? Can you make dendrograms based on cross-reactivity? But all of our databases are chemistry-centric. There's one compound per row, and they don't have a way to transpose that to be one assay per row and then have all the chemical properties. And there aren't good ways to search that. There aren't good ways to do similarity across the biological dimension instead of across the chemical dimension. We've had chemical similarity searching forever, but we don't. But even though it's the same data, we can't do the pivot of that. So we have to be able to have our chemistry department also be able to use a biologist's lens into the data.
That's a cool idea. Create a biological fingerprint to be able to look at different profiles. Yeah.
Yeah.
So actually, we have a few questions from the audience, but I'd really like to finish our themes because I'm not quite sure where these questions fit, and I don't want to take us off track. So we will get to the questions. Thank you for them, and keep them coming. So the next topic we wanted to address is leveraging AI, and we heard a lot about that today. So Woody, do you want to talk a little bit about how you guys leverage AI to improve your success?
Yeah, sure. I can kick things off, and I think that there's some questions that are related to this. So actually, we didn't use any AI bringing our Sting agonist into the clinic. We only made about 200 compounds in total. There were no known small molecules for Sting when we started the project. So that's just an example where it's unlikely that AI for the target would matter. The stuff that Eric's talking about and looking at off targets, obviously, that's a great application of better models, and I think the advanced machine learning models tend to be the best, even when the data is not that good. So, I think predicting off-target effects in areas where there are large quantities of high-quality data, which is rare, but at least in big pharma, they have a better chance at that. Public data sets are not good and not particularly useful, honestly. And I think more and more AI companies are starting to realize this. And I think data generation, high-quality data generation, is going to be a big part of the future. And I think that's really probably the biggest area for investment. We're starting to see it with some of the new companies that are coming out where their focus is really building a platform for data generation that will ultimately result in high quality predictions. I think as it is now, there's really niche areas for AI and machine learning. And so, the companies that say we're going to do drug discovery with machine learning, when you try to connect the pieces, it doesn't really make sense. But, for very specific applications, when you have the right data and there's data that's available either through, a big company or through public resources, it can be very useful. And it's just about being smart and, essentially continuing to do drug discovery the way that we've always done drug discovery, and then identifying potential problems where data can help make decisions and use that wisely. But it's by no means going to replace any of the other stuff for a long time, I would say. And I'd say most of the effort is going to be, most of the value is going to come through higher quality data sets. And that might be an area for an investment in terms of government funding as well. One could imagine a national campaign through NIH or European IMI and things like that, of just generating higher quality data, which I think would help everybody, and it would help all these startups who have brought in great talent from the tech field, really smart people, great programmers, but the data is ugly, and I think that's a problem for all of us.
Yeah, I'm actually really happy that was sort of how you introduced that. And I only say it from being someone who's had to sift through painstaking data sets and other things for antibiotics, drug discovery from natural products, where we as natural products chemists tend to be the worst when it comes to trying to name molecules. We have 27 different names for the same compound. And they're referenced in all different ways since the early 1900s. And it's quite the nightmare sometimes just doing the data cleanup. So I just wanted to at least say that I 100% agree with what you've said about that being imperative to the future it's good data goes in, then, good information will come out. So I definitely agree.
We do the same. We are trying to integrate machine learning and AI in the selection of our drug candidate. Again, the same problem. The data is so messy, and what's hard even is find the right data set. And if it's there out there, it's to have access to it, to analyze it in order to gain new insights. And this is all challenges that I don't know. I personally don't deal with all this analysis, so I leave it for the AI team. but they are struggling with it. And I wish we had like a magic solution for it, definitely.
Tisha, you wanted to say something? Did you? I thought I saw you.
Yeah, I was going to say, I think that it's great that you're really, bringing about the idea that the data sets are not very clean. And they are. I want to just say in the neurodegenerative space, there's a lot of efforts and funding to actually create robust data sets that go all the way from early discovery to clinical development. So that's a really important point that's being brought up, and it's being addressed. How quickly, that's a different story, but there's both private and public investments in that area to create those data sets that could be mined by any one public data set. So the AD model initiatives with very large funding through the Structural Genomics Consortium, it's going to be one that will handle a lot of data in the AD space. And there are other initiatives ongoing with Gates Venture, and so that's definitely recognizes a need, and hopefully will be addressed in the near future.
Yeah, and maybe if I could just say one more slightly more positive thing, to show the other side of the coin. There are specific areas, as we kind of alluded to, where it does make a lot of sense, and it's not necessarily these property predictions necessarily, but the recent progress by DeepMind and the AlphaFold 2 project using the PDB data, that made a lot of sense. A couple 100,000 structures, millions of contacts, lots of sequence alignment and multiple sequence alignment information, a really smart team and a very focused effort. And they basically, solved a problem that people thought was still decades away. So that's a great example. There's been some really great progress in cryo-EM refinement recently. Again, it's an image recognition problem, very well suited for machine learning, very large data sets, very hard to deal with manually, really falls right into the sweet spot of machine learning. So there's definitely been some great progress, but again, these are very narrow applications. Solving a protein structure is not solving drug discovery, as we've been talking about. It's very broad. So I think there's going to be these niche applications that'll be breakthroughs, and it'll help us overcome and move things faster. But across the board, this idea that we can predict drug properties and make better drugs, I think it's going to require a lot more data, a lot better data.
Go
ahead.
Eric?
Okay. Yeah. So, I think there are big areas of drug discovery where data really isn't going to even be the solution. I think that there are just a lot of things where experience and intuition are in areas that are sufficiently hard to quantify or to measure that it won't be able to be solved by machine intelligence until machine intelligence somehow manages to achieve that kind of intuition, if ever. And so I think that, again, getting back to cross-training and the fact that that people are taught within their disciplines, there's been a big rush of data scientists into pharma and drug discovery, and their mantra, I think, is in many cases end-to-end drug models that take just the inputs and a huge amount of data and produce the solution. But I think that the ones that are going to be successful are the ones that learn how to marry machine learning with human interaction and with intelligence, and allow the users, the experienced users that have discovered drugs and know and have learned those many lessons over many years the hard way, that they can guide the artificial intelligence as another team member towards finding a good solution that isn't just data driven.
I have a sort of a follow up on that too with what Eric's saying, is that it's always easier and cheaper to do it right the first time than it is to try to clean it up later. And that's something, again, I was addressing in my talk, is that the software has come a long way to being able to capture all the data that you need now and all the data that you may need in the future, but you don't know the questions that you have to ask later on. We have all the storage capacity you could ever want, right? That's basically infinite with the cloud. And then And the real issue, therefore, is all the policies and procedures, interdisciplinary areas we were talking about before, to make sure that we're capturing the right data in the first place. So if, because we're building the experimental data sets that now, one row at a time, that will be massive data sets that Eric will create cool models from, 5 years from now. And so that's really the key, I think, is, you do it right the first time, and then you don't have garbage data sets, and you don't have to clean it up later. And I think that's where the real trick is in trying to provide, software, again, on our side, that is a value add to the scientist as opposed to something that's getting in the way. There are so many times when I've gone into a lab and they say, well, this is the system we have now, and nobody uses it because it sucks. And therefore they don't collect data. They write stuff on paper towels and then move it over somewhere else later. I was just looking in the questions, and Kathleen Molnar was asking the question. Hi, Kathleen. Was asking the question about, software like Slack, right? How can that play a role in connecting teams? And, that kind of, productivity software that is boring from a scientific standpoint, but really key to how we just do day-to-day interactive work. That's the kind of stuff that if we can take advantage of it, and if scientists can, use it effectively in their workflow, that's going to make all the difference in the world. Create all your data sets up front, get all that data in there and get it good, and then you don't have to worry about massive cleanup projects.
Yep, totally agree.
I have a question about data. Do we believe there's a way to actually salvage all the data that exists that isn't clean enough to actually use in a meaningful way, or do we just say generate going forward in a particular format, standardized data, and then use that to actually build the data sets that are going to be used by machine learning models?
Well, it's not that there's no value, but the value is pretty low. So, might as well use it. It's better than nothing. But, it's not going to solve all the problems. And so, definitely going to require higher quality data. But machine learning is pretty amazing. I mean, even with messy data, and it looks like there's no signal, it can figure out, that this experimental, lab has an offset relative to this lab, even if the metadata is not included and you don't really know what the difference was in the buffer and the conditions, and somehow, fairly decent models still can come out. So I say absolutely use the data. It's better than nothing in most cases. But it's not going to solve the problems of drug discovery. It's not going to make it's not good enough to make drug discovery more efficient yet, and overcome all those challenges of how long it takes and how hard it takes, and the failure rate and all that stuff.
Tina, if I could chime in just with two things. The first would be a mea culpa on the behalf of academics. There's a lot of academic data that gets put out there that people have trouble producing. I think we're doing a better job than we have done in a long time with that, as the appreciation for reproducibility has really spread to the community more. So I think in the past you'd see a lot of probes that were published that were kinase inhibitors, but nobody had actually looked to see if they were selective or toxic. I think a lot of that's being rejected by journals now. I think editors are more sophisticated, and a lot of that is changing, and that's a good thing. To directly address the AI business, I would ask people to check out atomscience.org to learn more about the Atom Initiative that's going on at UCSF with GSK and Lawrence Livermore, where we're trying to leverage the massive computing power of the national labs, both at Lawrence Livermore as well as Frederick National Laboratory, and try and come up with some machine learning solutions to not figure out drug discovery, but accelerate portions of it. The first part of the program is taking a look at DILI, drug-induced liver injury, for which there's a lot of good data, and trying to discover ways we can predict that a priori from basically smile strings, from chemical structures, and then taking those predictions and testing them in a single lab using wet dystrophy. So creating that master data set using real data rather than cobbled together from a number of different sources using different assay conditions. So we're working towards it and trying to be part of the solution now instead of part of the problem.
Yeah, I would definitely say on even... Oh, sorry, go ahead.
Oh, no, I was going to say, I really want to, again, echo something that Jeff needs, and it's actually a project that I'm involved with, and it's called the Chemical Probes Portal, which is really using, cleaning up all that chemical biology data by using sharp chemical tools. So there is a portal that will actually make a recommendation. If you're going to interrogate a particular target with a chemical tool, they'll make a recommendation for which tool to use that is effective at the cellular concentration that's going to be used, which is a really huge problem. People still, use pan kinase inhibitors for specific kinase just because the literature has that as a MLK3, kinase inhibitor when it hits, 30% of the kinome. So there is a site called, chemicalprobes.org, and there are SAB or, key opinion leaders that will actually be able to guide you if you have a problem. But I think it's more and more, it's really important that we clean up the chemical biology literature so that it could be reproducible and people can actually make whatever decisions they make with those chemical probes to make better, drug discovery programs. It's kind of messy, I have to say. So it's a really important point that I want to echo on the point that Jeff made.
Yeah, and all I was going to add to all of it is, again, I come in from sort of the natural products side, and that we just notoriously have very dirty data. Comparatively to the pharmaceutical world or synthetic world, a lot of our compounds are kind of all over the place. But even on the receptor side, I'll say I at least hope it's getting better, but I guess I don't know if everyone has the same sentiment. But when it comes to trying to clean up some of that data, whether or not it's worth it to do that is a big question, because there's quite a few receptors now that we now know when they were testing them in the 80s or the 90s are actually 3 different receptors. They have different structures. Right. They're actually not the same. And so it is tough to really know, what was the actual receptor being tested, and, what do we actually know about that now? And then obviously on the protein structure side, that's a whole different ballgame. So I definitely agree a bit with Woody when he says it's hard to know how worth it is to clean up some of that sometimes. But I would love to say it's worth it, only because, obviously there's decades and decades of data that you don't want to have to toss, but it's hard. It's hard to know.
Maybe we can just start focusing on the current data, forget what's in the past.
I think it takes two sides of effort. One is from moving forward, the scientists, when they generating data, put in a high quality and make sure the format is easy to digest. But on the other hand, I think the computer science probably will need to figure out some way how to, salvage that kind of decades of data, because it's a human wisdom accumulation. Some of them are garbage, but think about AI. If you think it's a human, it's almost like a child, right? When they get into word, he doesn't know what is clean, what is not. You see a picture of a cat, you say maybe it's a tiger. But with time going, we see more and more picture of a real tiger and a real cat. You start to realize this is a cat, this is a tiger, right? So I think at some point with the technology development, we will be able to salvage at least part of those data. Discarding them is going to be a little bit waste of resource and all those time that a post of scientists putting their life into this research, right? So it definitely requires two sides to work together to help this field moving further.
It's funny that you mentioned that we have the cat and tiger. We actually have an application mentioned called Bioharmony Annotator. It used to be called BioAssayExpress, but it does assay annotation either going forward, starting from the lab, or from historic, from text documents. And we have a project right now with the Custodial Alliance about trying to verify, published data. And what's really interesting is there's some data where the system will learn really quickly what's a tiger and what's a cat, and be able to annotate it. And there are some publications where it's nearly impossible. And we actually have assembled some statistics about, these types of assays are nearly impossible to really sort of improve the quality of the data on. These kind of assays, actually, you can dig in a little bit deeper, and it's worth the effort to do so. So time and money for some things is worth it. For other things,
Yeah, even just the computer or the software will tell you, no, this I cannot do it. That alone is a good thing, because they will allow you to triage the whole body of data, saying, okay, this is worth mining. This is it's a lost cause, right? So even that part is, I think, is a progress for the field.
Yeah.
think a lot of what we think of as dirty data actually is biologically meaningful complexity that we just didn't or maybe still don't recognize. For biochemical assays that I work with, if two assays, medicinal chemists and I as well, in the past thought of the activity of my compound against my target as if that were some thermodynamic quantity like log P or solubility. And if two assays disagreed, then at least one of them must be wrong. And when I started actually doing multitask modeling and found that all of these assays for the same target gave very different results and started talking to the assay biologists, they said, this was kinases at the time, and they said, of course not, because they're different constructs. Some are just the catalytic domain, some are the full length. They have different degrees of post-translational modification. Kinases get phosphorylated, that affects their activity. You're inhibiting the ATP site, but it's phosphorylating a substrate. If you use a different substrate, you're going to get a different result. Kinases are regulated by forming multimers. And so if it's a monomer or a multimer in your assay, you're going to get a different result. Different concentrations of cofactors, all of these things change the environment of the protein, and then they change the result. And so you can't take one, you can't assume that an assay of compounds against the two different assays for the same target are the same. But the good news is that multitask modeling will through its algorithm, look at all of those multicollinearities between all of these different assays, and you build an assay you build a model for the particular assay that you're interested in, but the multitask model, all of these other assays, to the extent that they can, both by positive and negative contributions, and through these multicollinearities, are going to support that assay and improve its performance. And so you don't necessarily have to figure out all of these biochemical molecular details for those assays to be able to inform one another.
So we actually have some questions and comments by Steve Miller, who is very active in the question box. I was just looking over here that the discussion about bad data. He said maybe cleaning bad data is not a good focus of resources. He votes to look forward, anyway, appreciate all the comments.
One thing that Steve said, which I would like to touch on, is the idea where he says the open source world is now, and I totally agree with that. And we leverage open source data heavily. We have an open science team, with academics where we do pre-competitive, non-IP related work, and our view is that pharma in general has been too restrictive, and I know different pharma are more open to things, and I think it's actually evolving in a very constructive way. For example, Novartis and people like Jay Bradner that come in, and kind of see it's the challenge isn't about hiding things and keeping things secret in the old way that it used to be, but it's like we're all working on hard things, and if you don't get the best people and you're not working with the best technology and collaborating with academics, you're going to be left behind because the easy stuff is already done, and so it's no longer about this kind of secrecy and hiding things, and it's more about innovation, collaboration, and there's plenty of hard problems to solve and plenty of value out there. And if our goal is really to help patients, then it's about collaboration and open source. And I think Steve's right on with that one.
Right. I just have to make a comment. I hate to mention COVID, but you talk about collaboration and data sharing. That's why the vaccines were approved so quickly, right? For the first time, the regulators, industry, everyone is collaborating towards one goal. And so, shame on us if it takes us 10 years to develop drugs in the future, right? We should learn from this. So we've moved into the data topic. I know one of the issues about data that Rami had brought up was that we need to share the successful disease models across the industry. So this is another, just example of collaboration, that it's very difficult. Rami, you talk about this.
Yeah, and also I saw an example today, early this morning. It was someone from the Institute for System Biology or something like this. He did this presentation, and basically he collected data from over 900 patients, omics data from over 900 patients, just by doing collaboration or like having access to like public data. And this is allow him to basically identify critical pathways for the indication. It was like working on oncology and identify pathways that they are critical for this type of oncology or like this type of cancer. And they are working on targeting these pathways. And I think this is critical for drug development. And yeah, again, it goes back to the same, probably to previous topics, like how we can collaborate, how we can share data across the industry. I think that's a great idea, but on the ground, I feel this is going to be a little bit difficult, and we need a lot of effort and a lot of like be willing, specifically like big pharma company, a lot of will to do this. And I wish like instead of having in the Bay Area more than 100 or 200 biotech company, we have like just 50 that collaborate all together and like bring data faster. And like I wish that's like an option, but yeah, it's like a great idea, but at the same time, I feel it's very hard to execute. I wish I'm wrong, though.
Yeah, Rami, take a look at the Roivant model. They're pioneering this along with like Bluebird and a few other BioBridge. But Roivant in particular, kind of thinking about a financial model to, we're also scared of talking about money. We're just doing science, but money makes the world go round, and it's hard to do all the science without the money. And I think there might be some creative financial models that aren't going to be about like let's all just be nice and share, but it actually is going to be proven to be better to actually do this, and that you actually can produce better returns. You can get drugs out faster. We kind of have to start thinking about like how the financial system and the whole structure of the system can change. And I think that'll naturally force more collaboration, because that's going to produce better science and better drugs.
And I'd like to add that there are quite a few examples of that happening right now. So the epigenetic probes development from the Structural Genomics Consortium, that's a consortium that has about 6 different pharma at pre-competitive stage. Everyone files IP. They co-develop these probes, file IP, and everybody's got a whole IP kind of bag of tricks behind it. But there's this kind of collective working to nominate these compounds that are good starting points for everyone. And there's more and more consortia. There's, the top consortium is one where really we're, working across, academia, industry, and different institutes. And more these pre-competitive consortia are coming. And there's a lot of the open innovation portals for the pharma is just to do that, to generate this data so that they can, everyone can sort of elevate the platform, the starting point for drug discovery programs. So we're going to see a lot more of that, produce good examples of drugs in the next few years. It's clear that alone we don't do it. When you think about what, our history and drug development, right?
And I like what Woody said about the financial system. If there's something that's happening at the financial level, I think that collaboration could like happen a little bit faster and like Absolutely. In a clear, definitely.
I mean, when you think about all the wastage of parallel efforts that have been happening siloed within pharma walls, right? Everyone is working on the same kind of target development, target validation, assay development. None of that gets you any further, right? They're just kind of things that get you to some place. If you can come together up to that place and then depart from there, imagine how much saving you would do, because these are just kind of parallel processes that are happening in isolation, right? There's no IP, there's no there's really not a lot of values in these processes. So you could leverage that in parallel. I mean, collectively, it could be a huge savings for the industry in general, right?
Absolutely.
I don't think that's only just industry, also academic as well. Like, so many labs just working on their own, and the people keep trying to fail the thing again and again, and those things never got published, right? And speaking of publications, so many journals still not open access, and you have to pay to get the knowledge that, funded by government, funded by taxpayers' money, and you have to pay when you publish, and then you have to pay when you're trying to access the journal. I'm like, to me, that's not a good gesture in terms of openness and trying to share the knowledge among the scientific community. I think there is some government need to step in this, at least sharing journals freely in the scientific community, and then foster the scientific knowledge to share faster. And I mean, that's probably much easier than you ask two companies to share all their secrets and knowledge. Right? I mean, maybe I'm a bit naive here, but to me, I just don't understand. Even to this day, I mean, after 5 or 6 years after my PhD, I'm just being think why we need to pay to read the journals. We work so hard to contribute the work and publish them, and and we have to pay to read it. It's just doesn't make any sense to me.
As the business development guy on the call, I'm actually not afraid to talk about money. Even without paper, somebody does have to publish the journal and do all that work. So their salaries have to come from somewhere, which is why they charge money for them. I think though one of the points that's being brought up here that's really interesting though is that a lot of the collaborative work that can be done earlier on in the process, right? So if we have 10 years and $1 billion to discover a drug, the big money is spent later on in that process. This is later on in the process. Sorry, I don't know which side you're looking at. It's like looking in the mirror. Am I right or you're right? But the least expensive part of the process is earlier on, and I think that's a great time to collaborate, because if I was looking at the process and I can save 6 months early on for a lot less money than, it's like, hey, if I spend a little bit of money here and save 6 months, but I have to spend a ton of money over here to save 6 months, I'll spend a little bit of money to save 6 months and accelerate my time to market. So, I think this is like what you guys are saying. This is a good time to have these sort of early stage collaborations where, like you were saying, Leticia, we're all working on the same problem. So let's collaborate now in advance of when we need to kind of start going our own separate ways to make a buck a little bit later on. But it just makes sense.
Yeah, I definitely can say that for us being in sort of this suddenly booming psychedelic space, right? We are coming across a lot more investors that don't really understand our model, because we are focused on academic collaborations. We are focused on leveraging everything we can along the way, because why wouldn't you? I mean, it's the 21st century and we have all these things in our hands. We have easiest ways to communicate, than any time in history. And it just made a lot more sense to us to accelerate the early stage process. Like you mentioned, that's where you can really accelerate it and you can really collaborate a lot. But it is hard sometimes to explain that to those that are used to that more traditional model. They're used to the returns being based off of those more traditional pharma and biotech models. I think that, that is starting to change, but it's still out there. And I think it's harder to find those that are willing to be part of that change. But I mean, they're definitely becoming more apparent. There are more and more people out there who are part of it. You just have to search a little bit harder to find them. But I think it's worth the search when you do. And I think it's definitely worth the collaboration. It doesn't make sense not to anymore.
It looks like you probably need to see more success stories where that model actually generates returns for people and investors. And when that happens, I think more people are going to believe in that. But I just think, as you said, finding the right people who are willing to be open about this and get a couple of hits. Because at the end of the day, it's what he said, it's money that makes the world go round, right? So can't get away from it.
Unfortunately.
Yes.
Right. So why don't we move on, transition to another sort of related topic, and that is, another strategy that we need to ensure our success in drug discovery is a deeper understanding of our disease models. And I think Rami brought this up in our original conversation, that we need to understand things at a genomic level, and also talking about new cell-based models and other things. So, Rami, you want to start?
Yeah, I also think we need to understand like any preclinical model we use to ensure like the success in clinical trials, right? And somehow we are lucky. We have all this in vitro model, PDX model, iPSCs. So we have like all the animal models, but unfortunately, these models, they recapitulate partially the human disease. I'm not going to say all of them, but most of them. So this is why we need to understand a little bit the limitation, have like a deep understanding of each model. What's the limitation? What's the aspect that resemble the human disease? What the aspect that does not? And like try as much as we can, like to increase the number of models we are using to basically cover each aspect of the human disease. So we're like have stronger data and like we're more confident when we go into clinical trials. And this is something I think all of us in the industry are doing it. Having multiple genetically engineered animal model, having multiple cell lines, and when it's available, definitely PDX model is and iPSCs. That's like that would be like a powerful tool since like they are originated from the human disease itself. So this is something great. And one thing also I feel like sometimes it's missing in the preclinical analysis is the outcome of the disease. I really thought that the outcome measure should be reflective or like should be at least of a proven relevance in a clinical setting. And in the muscle industry, for example, in clinical trial, we have like a standard primary outcome, which is 6 minutes walk, or like the capacity of the patient to walk for 6 minutes, like the distance walked by the patient. And when we go to the preclinical data, we focus a lot on muscle mass, or like increased muscle size, and they are not related. And like a lot of drugs failed when it went to the clinical trial. And I think like we need to include all these biomarkers, all these outcomes, are like primary outcomes from clinical trials in our preclinical studies.
Interesting. Yeah, that would be really a challenge in some of these models, like the muscle models.
Yeah.
And I think, I mean, we're fortunate that we're actually now there's a few precedents for the FDA to accept data and approvals without the need of animal models. So I think it's being very specific about the disease, have your target engagement, your PD response, to know that you've modulated your target. And then if the data comes from an iPSC, patient-derived iPSC, or some very disease-relevant organoid data, it doesn't have to be, animal data, as long as you have all of the, safety, pharmacology, and everything that you need from, that you need for your IND package. But I think we need to start thinking beyond the use of animal models, because most of them are flawed in some ways or another. They're transcripts issue. I mean, they're sort of issues with some of these models. Really, very severe transgene issues, right? That some, you follow them and they somehow recapitulate the biology, but then they don't have anything to do with the biology. It has to do with what transgene was. So I think more and more we need to look forward to disease-specific models that are relevant to disease, and that get away from these early rodent models that don't, at least for in case we work with brain diseases and we're still tied to rodent models, right, and that don't really represent what's happening.
Right. There's certainly a lot of activity in the bringing, human cells, human tissues, the human factor into these animal models, whether it's knock-ins or these constructs, these 3D constructs using human cells, the organoids, spheroids, and things. I think that certainly has a lot of promise.
Right.
Yeah, and there are humanized animals as well that may be helpful at some point. Yeah, I mean, definitely the species between species transferability is a big issue. Like in our field for delivery, in particular, for example, lipid nanoparticles, we found some of the formulations works extremely well in rodent, and when we put them in monkeys, they're totally opposite. Like some of bad ones work so well in monkey, and then some of the stellar ones in rodent works just not so great in NHP, and God knows how this kind of NHP going to translate to human. There are so many things we have to pay attention to when we do our lead optimizations, etc. So it's very important. I would say even the $1 million questions in the drug development process is to find the right model and a good model.
Right, and the approach of many different models and putting comprehensive information together, I think is another approach that we can't just rely on a single model. Go ahead.
Yeah, definitely. I think each model has will deliver a specific information, and it would be nice to have multiple models. As you said, like probably rodent, but also go to primates, or like go to bigger animals, or like use PDX models or iPSCs. So we should be like more aware. I think we are kind of aware, but like sometimes we try to forget it, or like not pay attention enough to it. Be aware to the limitation of each model, and like how we can compensate by using another model to answer a specific question. That's like part or like a component of the human disease.
I guess we're conditioned historically to think about choosing a model that will give us the answer that will let us take the molecule forward, right? Or kill the molecule and just be done with it. And people don't really look at this as, okay, let's try to make sure we get all the data we can from, as you said, multiple models to get a true picture of what the molecule is doing. So I think that conditioning also has to change a little bit in our mindset and how we try to approach this problem. I know from when I was doing this, it was always, what's the model that's going to give me the best shot at actually making a decision on this molecule so I can do something with it, right? Yes or no, and then move on from there, because you want to move quickly and move fast. So I think it plays into what you're saying, not being afraid of the different data sets you're going to get from different models, and understanding the power of the totality of the data you get from it.
Right. So why don't we transition to another theme? And this is actually, Letitia, this is from your graphic that we actually showed before you arrived today, about your systematic process of taking your IP from academia to regulatory approval, and how you do gap analysis, and you've got transition criteria for every step. You make sure that you have all of your steps covered before you move to the next step. Would you like to expound on that?
Sure. I'm sure that most of the my colleagues in the panel do this already, but it's just really important to repeat that often really basic questions like, is your target in the tissue that you're targeting, right? These questions are really important to ask, right? And often we take for granted. I think, Eric, you really, when you were talking about this sort of context specific of this assay, I think that's really critical. When we try to compare cellular data, these are state-dependent data. If you're looking at kinase inhibition, they're going to depend on what cell type, what kinome is in that cell type, what conditions are applied to those cells. So really making sure that as we move from each one of these steps in the drug discovery kind of the pipeline, that we really ask those critical questions to ensure that we understand the data we have And are we ready to move on to the next one? Do we really have an understanding of what data we have? And in some cases, we don't need it. We don't need to have everything, but we really need to know that what we have is really solid before we move on, because it's expensive process, right? So the more you can ask at the earlier stage to make your go no go decisions, the better you are, right? And we definitely know, we know that it's very expensive process. So the earlier you can kill that program, if you need to, if you have target based toxicity before you run. Same thing with hits. Validate your hits. I'm very I don't like to call them even hits before they really get validated. So they're called actives, and then you have your validation, then you call them hits, and you move on to hit to lead. But really take the time to do that process so that you can eliminate things early. I think this is, I'm echoing a lot of things that we all do, but it's really important to have a real, very robust decision-making criteria to move along that pipeline before you spend a lot of money.
Yeah, it's something we deal with a lot in the Small Molecular Discovery Center at UCSF, where we have PIs that come in that are very, very pure academics and have the idea that they'll screen a 5,000-member library and then go straight to the clinic. And the idea that not everything in there is pure gold, and not all actives are hits, and not all hits are created equal, and some things are not optimizable. Some things are pan-assay interference compounds, and kind of guiding people down this path is a lot of what we do for academic PIs, as well as some of the people that come in from small companies that, again, have this idea that you can go very, very quickly from the screen to phase one. And it's not in our experience that happens.
Right. I get that a lot as well. And, the other point, too, is that all drugs are, coming from different places potentially, right? If you're in licensing a drug and maybe you didn't do the due diligence and you've got to go back and look at the data that's there, you've got to decide, do I have confidence in the data or do I need to repeat it, right? So, all drugs aren't created internally. Right.
If you're asking that question to be repeated, you got to make sure you do it. Even for projects that we bring in internally, looking to fund things internally through innovation, that data gets repeated. You just have to do it. I've been burned too many times. You all have been burned too many times.
Also, I think this is like a good place to use the help of CROs. Like if we can repeat this experiment using CROs, that I think a key to start like considering this molecule as a hit, or like considering this molecule as a future hit. Because if all the data are generated internally, yeah, that's like probably going to be like problematic. But like if it's also an external source is able to produce the same data, that would be powerful.
I think, speaking for the CRO now, where I am now, I clearly completely agree with you, because I think all of you should be doing that. but it's also true from the investment perspective, right? Because I think investors also want to have that independent verification of the data being robust, repeatable, reproducible. And then I think one of the points that we talked about collaboration, it's a point that I had raised in our earlier conversation, is I think at many of the CROs, the level of expertise that's now available at the CROs, because of the variety of things that they work on, is so massive and so extensive. You have more available there, and I don't think that everybody taps into it to the same extent that they should. And being able to kind of be a little bit more open about what you're actually trying to do and what you're driving towards, you can leverage that skill and that knowledge and do a little better than you might if you just said, go make this one for me, or run this assay for me, or just give me the data for this. So, but definitely believe that that's very true.
Yeah, I think the reproducibility thing is even big on a very basic level when it comes to just the environment you're trying to do something in. So I live in Florida. It's hot and humid. Everything's wet. Trying to do anything dry is just a nightmare. And suddenly, you try to do it somewhere else. Maybe it works great. Maybe it's worse, especially if it's formulation, if it's stability, if it's anything. I mean, it can be completely different just because of where you're located. And I think that those are really, they seem so simple, but are so important for if you're actually trying to then mass manufacture this product on a drug level pharmaceutically. And so I definitely think reproducibility in multiple locations is ideal.
That will requires attention to a lot of details in terms of the experiment and the process. I mean, what I learned is in the industry, you're sort of trying to record everything in a very detailed and accurate way, but in academic, like, hey, I would do it, and maybe let me think, it's 1 mice or 2 mice, maybe not exactly the same, right? So I mean, it definitely requires a whole body of scientists to do the same, just trying to record what they are. I mean, a lot of times I find a paper with reference to antibody, and I find it's not even like the right one to even use for that target to be detected. I mean, that's a lot of details need to be recorded accurately to make things can be repeatable. And this is and like talking about what we just speak of is that reproducibility, and also not repeating others' errors, and that also requires a very accurate record of your experiment conditions, and have all those details taken into account.
Mm-hmm.
Right. Any other comments about that? I think in the last 15 minutes or so that we have, I really want to address the theme of customized approaches needed, and that we've sort of talked about this a bit in different stages of drug discovery, but the customized approach that's needed from the beginning all the way to the end, right? The things that impact you with marketing and competitors, and are you going to be able, if you're in the rare disease area, are you going to be able to find patients for your clinical trials? So the practicality and feasibility issues is not all about science. Anybody want to comment on that?
Yeah, maybe I can get started a bit on this. I mean, this is probably a common theme of the whole process of drug development. From you identify the indications, from you try to find the target, when you do the target validation, selecting the model, for example, and then regulatory submission, and determine your targeted product profile or TPP, and also the clinical trials. It's a common theme throughout the whole process. I mean, it seems pretty common sense, like the drug development will take all these, but it may be not that obvious. Like each niche or each step, it needs to be highly customized to the disease, to the target, to the even drug modality. For siRNA, mRNA, and the small molecules, they're all different. How you design your experiment, how you design your study will be very different in terms of all these different aspects. And I'll give example in terms of drug like a disease indication. So, for example, infectious disease, it has a very straightforward etiology of the cause of disease. It's a viral infection or bacterial infection, and usually your strategy will be, okay, I'm wanting to antibacteria or antivirus, and you have much less to do with your host proteins, and you have lower risk of affecting your host, normal host physiological functions. And then for other disease like cancer or NASH, which has a much more complicated mechanism of pathogenesis, then your development strategy would be much more different. It's not saying like, okay, I'll just put a virus in a cell and let's try to kill the virus. That's it. And for NASH and cancer, it's very, very different. You have gene mutations, you have epigenetics, you have metabolic issues, and everything would be contributing to the complex of the disease. And that is why, like, for some of the complicated disease, why it is so difficult to tackle. The lower hanging fruit is already being taken by others, and it will take a lot of effort to understand the disease first before you go inside and to find the right treatment. Just like Jeff was mentioning, like, okay, I just have this 5,000 or 50,000 compounds and I want to cure cancer. It doesn't work in that way. You really have to understand disease first and do the data mining. Maybe even AI or computer science will come in at this stage to help you to understand everything and start to work on it. And also, in terms of like rare disease with versus common disease, your TPPs in terms of like how many patients you want to have in your clinical trials, and also risk benefit ratio or profile will be different. For some like life and death disease, patients are dying in the bed, you won't care like, oh, I will have a little bit fever as a side effect. But if you say I want to treat the hypertension and even a little bit cough, because my parents have like hypertension, and I understand like if my doctor says, oh, I'm sorry, this drug works perfect, but you may have a little bit cough during the night. It's not a big deal, but I would say, can you find me a better one? So it's all like you need to really customize and think about, okay, when this drug hit in the market, whether the patient is going to like it, whether really going to help the patient, and whether the payer is going to like it, whether the government is going to like it. So there's so many factors to take into account when you at the first stage sitting at the desk thinking, okay, I want to develop a treatment. And then the question is what kind of treatment and how good you want to be and what kind of patient you really want to help. So it's a lot of things you need to take into account.
Right. And and Jin, in the rare disease space also, considering the competition and where you are, in the race for a cure, so to speak. And so, is your competitor going to use up all of your
child patients, right? Yeah, right. Exactly.
Yeah, well, rare diseases is interesting too because you get that priority review kit that you can use for something else. So if you have a mechanism that's useful in a rare disease that has a well-established patient population to tap into for a small trial, that could be a really good way to get a first approval for a drug with a relatively inexpensive trial for a small group of people, and you get that priority review chip from using the rare orphan drug designation. So strategically, that's an interesting option also.
Yeah, but that definitely requires to have a full understanding of the competitive landscape, right?
Absolutely.
As Tina was just alluding to, like, if like 5 other companies also working on this, like 1,000 patients, and by the time you hit the clinic, they already take all the patients. They say, okay, I have to wait another 20 years to gather all these patients for phase 1, or phase 1B, or phase 2. Like, sorry, also, that's not an option.
We have a great example of the ACE inhibitors too, and the ACE cough that comes for angiotensin converting enzyme for hypertension. There's a case where that population is pretty well served, right? There's a lot of good medications there. So if you're going to be developing a medication for hypertension, it's got to be darn good. And it's going to have to be cheap. And that's a tough space to break into. Whereas if you're looking at something like stage 4 lung cancer, where there's not much out there that's going to get you past 9 months, there's more room to operate there in a large patient population. And you'll see, we saw Keytruda approved for non-small cell lung cancer for people with wild-type EGFR. And there's that room. There's room in that patient population to carve out a niche for the trial, as well as people that are willing to take this drug because they're relatively underserved by what's out there already.
Yeah, I'm really glad that you brought in the perspective of thinking about what patients will actually want to use. And that's really just something that I think a lot of people don't really think about along the way. I mean, I'll admit, until I got into the space after grad school and postdocs and things, and I actually realized, there's a world outside of like the lab in academia. I remembered, wait, like someone has to actually want this. Someone has to actually think to themselves, I will use that and it will benefit me and I'm okay with it. I mean, a lot of times we get so caught up in sort of the minutia of every day in the lab and the process and trying to figure out the best, cures along the way. But I think that's just a hugely important aspect of it that a lot of times is left sort of off to the side. And it's sort of said, maybe we can just make an IV instead. And it's like, there's a lot of people who don't like that. there's a lot of other options now. So I think it's very important.
And it's hard to get reimbursed too. You have to think of the managed care side of it too, about what's going to be reimbursable down the line.
Yep.
Right.
Our site focuses on neglected diseases for the third world, and there are some really unusual constraints, like how valuable it is to have a drug which would be a single injection cure for the disease is ideal, because it might be that the patient has to walk 30 miles to a clinic, and if they have to do that twice, that would be really a big burden. And if you give them pills, say it takes 6 pills, well, they might take 2 for themselves and give 2 to their mother and give 2 to their sister, thinking we all have disease, maybe we can get heart cured or something like that. And so that, the idea of a single injection being the ideal route of administration for for where we live, that would be, pretty crazy. Also, there are indications for particular compartments, like cryptosporidiosis, it affects the insides of the intestinal membrane. And so we work so hard to learn how to make oral systemic drugs that are highly bioavailable. But here, ideally, you have a drug that never gets past the internal membrane of the enterocytes where the parasite lives. And so you think about things like, well, maybe PGP export is a good thing.
Yeah.
You need to get some permeability, but you don't ever want to get to, the portal vein. And another example of that is drugs for radioligand imaging, where their properties have to be completely different than what you would normally do for an oral therapeutic. And it becomes fascinating because as medicinal chemists, that's so contrary to the way we normally think about how do you design a drug, and the rules are entirely different, and we don't really even understand them because there are so few examples.
That circle back to the interdiscipline of at the beginning, you not just to sort of okay, I understand this thing. You have to understand the whole part of the disease and what is really needed as a drug product. So that's why you drafting a TPP at the beginning of your project is so helpful, because you're going to put that in front of everybody who's going to be involved to say, okay, this is what I want, and this is the goal I wanted to achieve. So everybody going to work together to that direction.
Yeah. I mean, one interesting point is it's easier to do that when you have benchmark drugs. It's really harder when you're going into new indications in rare diseases where you don't really know what that looks like. So it's it's a bit of a moving target. And I want to bring up a point that was made earlier about sort of target populations, like what is what is a target population that you're going to test your drug on, especially for rare diseases. And I think it's important to kind of point that there's new ways that are being solved, that these problems are being solved, like platform trials where multiple companies are coming together to test their compounds on multiple arms of the same kind of cohorts of people. So there are a lot of really interesting, new solutions that are coming up to solve these issues of rare patients, for example, and that's one of the things that we're constantly struggling with. If we don't have drugs there yet, but when we do, where are we going to test them? Do we have enough patients? Do we have to go beyond where North America, go to other places where these diseases might be prevalent? So it's really very important things to look at very, very early. But again, echo the point is we don't always know what we need to end up with. We sort of have an idea, but it's kind of good to be flexible to know that, we might need to adjust with that target product profile might look like.
Right. So Jackie, I'm thinking that you must have some interesting perspective about patients and clinical trials and the people who are going to be interested in buying your drugs when they get there. I mean, I guess you guys are really early, right? So maybe you're not thinking about.
So I will say, before I really switched gears into this industry, I did work in the cannabis industry for a few years in more pharmaceutically focused types of companies and more on the medicinal side. But it was the first time that I was able to experience both working in manufacturing and the lab and product development, and then being able to go to a dispensary and directly talk to a patient about what they thought about our products and how it actually worked. I had never been able to be part of that entire process until then. And after a few years of that, you really learn how finicky some patients are about the littlest things. I mean, really things that I would never think are a big deal, but they are to other people. And those are obviously not clinical trials by any means. They're not controlled necessarily. It's just talking to the people about what they're using and how it affects them. But I will say that now going into the more regulated space and actually going to more pharmaceutical space of bringing the drug to the market and something that's similar to a psychedelic scaffold, that's a huge focus of ours, even if we're not going to be part of it towards the end, is what outcomes we want and what patients will actually use. And, as Letitia mentioned, we have to be flexible. We have to be, I always think that that's important along the way, but I also know that if someone is taking a large dose of LSD with a needle in their arm, you might have some not so great outcomes with that person, looking down at their arm and suddenly, oh, what's going on? And I think that there's a lot of unknowns about what patients are going to experience, how that it's going to look. And I mean, we just don't know a lot about, the outcomes of these things. So I think we are very aware of all of that, even though we are still very much preclinical. We know that there are a lot of different things that could make something seemingly go wrong quick just because you didn't think about it ahead of time. So it's definitely on the forefront of our minds. Yeah.
Very interesting space.
Yeah.
Some people who are in your space are, get their anecdotal data from other countries, right? So they're not even able to do what they need to do here yet.
Yeah, our focus is definitely, we're one of the few that's within the U.S. And I mean, we have collaborators at the University of South Florida, as well as Wisconsin and the National Institute of Drug Abuse. We're trying to, work with whoever we can along the way who's had any experience in the space, because there are people who have been doing this for a long time. I realize there's a lot of companies that want to tell you they're the first to do everything right now in the space, but there have been people doing research on this, and it's worth working with everyone you can along the way and making sure that we do it the best we can and make sure that patients benefit from it. I think that at this point, we all know that there's benefits. It's just finding the best route of administration, finding the best way to bring those benefits to patients, and finding the best targets. So,
right. I think it's a great opportunity. I was doing rat studies on ibogaine back in 1993. And that was incredibly exciting at the time. And it's great that, the industry hasn't given up, that people are still pursuing it.
Yeah. And that's obviously a huge one for, substance use disorders. And I will say that a lot of attention is not always given to alcohol abuse, even nicotine addictions, obviously still a thing. The opioid, epidemic that happened is on, everyone's minds. But, even now with COVID and everything else, there's a reason that I think in the New York Times, they came out with it after the first couple months of the pandemic, just the escalating sales of liquor stores and alcohol and everything else. And that's not even including the things you're never going to be able to document from, underground markets and other places. So there's a need for not only the substance abuse, but also just mental health. We all know it. We all have someone that's either, if they're not directly related to us, they're maybe once removed that is struggling. So it's worth it. It's not going to be solved overnight. Nothing in drug discovery is ever solved overnight. And this is drug discovery. It's still in that space, even though we know some of the outcomes. We might have decent safety profiles, which is great. But it still requires a lot of research to make sure that it goes the way it needs to go to benefit people. Yeah.
Anybody have any final thoughts they want to share with the panel or the audience?
I'd say one of the most biggest insights from the chat that wasn't discussed was people should be using Slack. Phenomenally useful productivity tool. And I hope it doesn't go south with Salesforce converting it into some kind of sales agenda.
Yeah. So I guess I'll close. I was listening to Dr. Ranga's talk this morning, the guy from, is it Berg Health? And I thought he had a really profound and hopeful quote. He said that the gap between drug development and the management of health is closing. So I thought that was really encouraging, and we don't hear a lot of encouraging things about our industry. So I really appreciate his positivity on that, and I wish you all the best of luck in your in closing your gaps.
Thank you.
So I guess
Thank you.
thank you all so much for participating. It was a great panel.
Yeah, thank you.
Thanks, everybody.
Okay.
Thank you.
See you tomorrow.
Take care.
Bye.
Bye.